Good day everyone, welcome to the course on biological data analysis and visualization with R. In the last class we have started talking about basic visualization techniques. We have seen how to generate histograms, density plots, etc. And then we talked about a data set for mean SD plot. So, we will continue with that in this class, right. We will talk about more data visualization, different kinds of plots that you can use and that are very useful for biological data, right. So, this is something we will be talking about in this class. So, the first topic is we will generate this mean SD plot, the data set we have already introduced, we have seen a little bit in the last class. So, we will generate the plot and see like how the data looks like, right. This is something that is the purpose of visualization. We will then talk about box plot. Okay. So, box plot gives us again a different way of representing the data, but let us say we want to compare the distribution of values of multiple parameters, right, or of a single parameter observed under different conditions. Okay. So, box plot is kind of a very nice way to represent this, because box plot will give us the median value, the interquartile range, etc., and we can also show the values, individual data points on top of that box plot. So, we will see in a moment how this actually looks like and how we can compare a value of a parameter under different conditions. Okay. And we can extend this to not just 2, but maybe 4, 5, 10, as many as you want. Okay. And then thirdly, I will just very briefly mention violin plot. So, sometimes we use violin plot as an alternative to box plot. It is very similar idea. The only thing we have is that violin plot actually shows how many data points are there at each kind of value, right. So, again we have these values in the y axis, and x axis is the different the parameters at different conditions, and we want to get the distribution of these parameters, right, and or visualize these distributions. Violin plot gives us the width, has a width, right, which is proportional to the number of observation for each y value. Okay. So, we will see this when we generate these plots. I think that would be clear. Okay. So, let us start with the mean SD plot first. So, we will see the mean values and then followed by we will add the error bars that will show the standard deviation. Okay. And we will start with that first, and then we will talk about box plot and bioplot. So, the box plot is we will use the command box plot, and there is we will also use the command strip chart. Okay. So, strip chart command is actually used to add the data points. Okay. So, we will talk about this in step by step manner, and finally, I will just mention the bioplot command. So, this is part of a bioplot package. The earlier commands, whatever we are using so far, the hist command or the box plot command here, all these things are part of R, right, standard R commands. Okay. Whereas, for bioplot command you have to use this package bioplot and load this package. So, first install this package and then load it. Okay. So, let us now go into this. So, we will start with the mean SD plot first with the data set that we have introduced already, and then we will follow that up with the box plot. Okay. All right. So, let us go to R, and here we have the data. Okay. So, this is the data set we have seen in the last class. Okay. This is a growth curve data, and I showed you, right, this is how the data looks like. We have time in one column, in the first column, and then we have these different measurements and different conditions, right. So, we have G01 YPD1, G01 YPD2, etc. So, these are replicates. So, G01 is one strain, it replicates 3 replicate measurements for this. Okay. So, since we have these replicate measurements, we want to calculate the mean and we want to represent the standard deviation. Okay. So, this is a very common type of data that we have in biological data, right, and we have this for multiple strains here, and we also have the time value, right. So, we have a huge number of points in here. Okay. So, what we will do is we will take this data. So, we have observed this for 24 hours, right. So, we have a lot of points and for each of these strains, okay, and the replicates. So, what we will do is from this data table we will calculate the mean value as well as SD value, and then we will generate the plot. To do that, we will use a package, right, which can actually generate this mean and SD. The package is called matrix stats. Okay. So, let us see if this package is installed, if otherwise we will install it. So, it is there, In your system, if it is not there, please install, right, again using this command install.packages matrix stats, okay, because we will use some functions from these matrix stats packages. Okay. So, we have already loaded the data. So, this is the R code that we have, right. So, again I will share this code with you for so that you can try this out yourself. So, this is the matrix stats package we have loaded. We have loaded the data already, it is in the tab variables. We have seen the data. What we are going to do is we want to calculate this, right. Now, we are using these functions from matrix stats package, the row means and row SDs, right. So, we want the mean and standard deviation for each strain at each time point, right. So, for each row we have to calculate these means of each 3 replicates for each strain, right. So, this is what we are doing here, and if you remember, right. So, this tab C234, what does it mean is we are looking into the column 234, right. So, we are taking the columns 2, 3, 4 of this data table, and we are calculating the row means, okay. And similarly, for row SDs, we are taking this same data table and we are calculating these row SDs, okay. So, this is something that we will do, right, and similarly we are doing this for all the strains that we have, okay. So, let us calculate this. Let us try this first for one strain G01, okay, and let us see what are the values we have. Okay, this is head and we also get C0SD. Okay, so, we have the mean value and SD value for each row, right, for each time point we have these values now, okay, and we have calculated this for G01. So, you might ask like, so these row means we need that we can directly run on this tab, but for row SDs we have to store these as metrics, and this is something that is required, right. You will see when you run this, this is something that will be required by the commands. So, we have done this for one strain. So, let us do this for other strains one by one, right. So, we have for G02, right, we will run this here. Okay. So, this is something that we have seen from the data, right. So, G02 is in column 5, 6, 7, right, and that is why we are using this, we are accessing those elements and we are calculating these row means and row SDs as we have done for G01, right. So, we can simply repeat this process for all of them, right, and we know that because we know the columns where these replicates are present. So, we have this G03 mean 8, 9, 10, G1001 mean 11, 12, 13 and so on, right. So, once we do that, okay, so we then generate all the mean and the SD values, okay. Now, for this plot what we have to do is we also have to use these mean and SD values to actually generate the upper value and the lower value, right. So, you have this standard deviation that will be given as error bars. So, you have to given the generate the upper value as well as the lower limit, right. So, upper limit and lower limit. So, which means SD sorry mean plus minus SD, right. So, that is what we are going to do now. So, here that is what we are doing, right. In the first place we have Z01 down, which is Z01 mean minus Z01 SD. So, this is mean minus SD and the next one is mean plus SD, right. So, we are generating this mean plus minus 1 SD, okay, and we do this for all the strains. So, for the first strain we have done this Z01, for the other strains we follow the same process, okay. Okay. So, this is something now we have done, okay, and we will use this information, these values that we have generated for generating the plots, okay. So, how do you generate this plot? This is something I will discuss now. Here is this is the code, okay. So, we have to use the plot, right, and first we will plot the mean value, right. So, this is a simple plot xy, right. So, our time is x axis and Z01 mean is the y axis, that is the growth, right. So, this is simple xy plot, okay. So, that is what we start with, followed by you see these parameters which you are now familiar with, okay. We have introduced all these parameters, right. So, what are these parameters is PCH 20 is a parameter for the points, right. Then you have the size parameters for axis, label, as well as this CEX 1.2 is the parameter for the points themselves, right. The plots that we are generating, the points in those plots, those size will be controlled by this CEX parameter. Okay. So, size of those points, and then we have the size of these fonts, right, for the axis and the label, and we have the x label which is time, right, and y label which is only 600. Then we have color. So, what is this color? We have used red, blue, green, etc. So, in R you have these codes for all colors. So, you can simply search R color codes and you will see these codes, right. So, we are using these colors because then now we can access very different shades of colors, right. So, this is something what we are going to do. Okay. So, I will simply type this and this is what we have generated. So, what you see is we have said x, y, right. So, x is time, y is the mean value, right, that we calculated before, and then we have the PCH which actually determines the point types that we are going to use, and then size of the points, etc., and the color. Okay. So, this is color, this is dark blue or navy blue color. Okay. Then what we want to do is we want to add line to this line through this color, right. So, line through these points of same color, right. So, this is what we are adding this line, right, again line x y and this color. So, we want the line of the same color, right, and this is what the line looks like. Okay. And then we want to add the standard deviation now, right, mean plus minus SD. Okay. So, this is the command that we use for adding this plus minus SD, right. The command is arrows, okay, followed by what you have to give is the you are drawing these arrows or these lines, right, the mean SD lines, okay, using this command arrows, okay, between these x y coordinates. So, where you should where these arrows should start, okay, the x y coordinate where it should start and the x y coordinate where it should end. Okay. So, for each time point, right, the x coordinate will remain the same, right, x coordinate is not changing, the standard deviation will be from some y value to some higher y value, right. So, that is the mean minus SD and mean plus SD which we calculated already, right, this g01 dot down, g01 dot up, right. So, those will give us the y coordinates. Okay. So, first we have to define this x y coordinate from where this arrow should start and the x y coordinate, right, where this arrow should end. Okay. Then you have this chord equals to 0, angle equals to 90. It actually shows the orientation of this arrow, how this arrow should look like. Okay. You can try this out, right, changing this angle and changing this chord to 0, 1, 2, etc., and you will see that this will look different. We want that these arrows of standard deviation, right, in same color as the plot, right. So, we will use this color, same color code, and then we have also the size of the arrows. Okay. So, this is CX, this is size of the arrows. Okay. Let us do that, and we have added the arrows, but of course, because the standard deviation is very small. So, they are not visible here at this point. Okay. So, you can try this out for other data points where you will see that these will be visible, okay, where you can see the standard deviation. We will now add this for other strains. So, this is for G01, we will add this for other strains, and you will see for some of them we will be able to see the standard deviation, right, this mean plus minus SD. Okay. So, now we want to do this for the second strain, okay, and on top of this plot, okay, not another plot, because we want to compare this all these 6 strains in one plot and see how they are growing, right, whether they are growing in the similar manner or whether there is any difference. Okay. So, we use this command now, we will not use the plot command anymore, we will use this command points, okay, and it is very simple now. Is with this points command we have the x axis, the x values only, and the y values is the mean values, and then we say what should be the point type and the size of these points and the color. We do not need to define axis anymore because we have already defined those. Okay. So, I am adding those points, they are in lighter shade of blue and they are kind of just are on top of almost similar, right. So, to the other one that we have seen. Similarly, for this one we also want to add a line. Okay. So, in this case, okay, they look very similar, right, almost identical. So, maybe you might not be able to see, and we add the standard deviation. For some, maybe if you look carefully, you have seen these standard deviation lines appear for some points. We will see this better when you add the next few ones, right. So, we do the same thing, right, similar kind of plot for the next strain, right, and this is even lighter shade of blue. Again, this they are kind of overlapping with each other a lot, right. Maybe let us try the other 3 strains and see whether our luck changes. Okay. Yeah. So, you see this now, this is in darker shade of red and or brown, right. You can see this is different from actually the first 3 strains, and here you can see the standard deviation here, right. So, this is actually due to this code and the angle, right. So, this if you can change these parameters, right, these arrows, right, what is the angle and the code, you will see these mean plus minus HT, they will look different, right. There will be caps, without caps, etc. You can try out this. Okay. So, you can now see this standard deviation here, at least for this strain. We will try out the next 2 similar way. So, I am just copying this, you understand the code, and this one also is different, you can see this, right, and the final one is this one. Okay. You can now see the some difference, and you can see these error bars at least for some strains, right, and you can now probably say, okay, some of these strains, the ones in red, yellow, they are actually different from the blue ones, right, they are showing some difference in growth characteristics. Okay. So, this is something that is good, right. So, we have kind of identified this, and we can store this data and we can save it again using the same command def.copy to PDF, right, and the file name, right, let us say we are saying growth curve GUIPD. We can also use these width and height parameters when we are saving as files. So, it is like how wide and high this file should be. So, this is kind of like determine the file size. Just be informed that this width and height parameter might vary based on our version that you are using. So, in earlier versions these numbers used to be different, but in the current version it is usually these numbers that we have to use. So, you have to try this out, right. In earlier versions we used to use like 900, 600 kind of scale, here it is different. Okay. So, the same the rest of the things remains the same, we use the def.copy to PDF. So, it has been saved, we can say simply def.off. Okay. So, this has been saved now. One thing I will also mention, and you will probably see in this file as well, and this is something that is good practice, is that if you want to now on the same R console you want to load another data and run another type of analysis, it is actually better to clear the environment. Okay. So, let us say what we have in the environment, right, these are the parameters that are there in the environment, right. So, it is better to clear this environment by saying RM. Okay. So, when you say RM tab, it means we will clearing the environment, there is no object as tab anymore, right. So, you can see this tab object is gone. So, that is what this code is doing here, right. So, this RM code is actually removing these parameters from the environment, because otherwise they might interfere, they might actually by mistake you might use the same name, and in case your operation was not successful, it will just take the value from the previous analysis. Okay. So, it is always a good practice to actually clear the environment after you are done with your analysis. Okay. So, this actually reduces any chance of mistakes, etc. Okay. So, we have seen the mean SD plot and we have saved it, okay, in a PDF file. The next one what we are going to do is box plot. Okay. So, we have saved the growth curve here, you can see this. Okay. So, this is how it looks like. This is because of the width and height parameter that we described. So, that is why we have this kind of shape. You can change it and the whole thing will change. Okay. And this looks most appropriate for this growth curve. Okay. So, the next one we will use is the box plot. Okay. So, as we said, we want to discuss box plot, and now we will go back to the original data, right, the noise data that we are using, right, and you have seen this now many times. So, that data set is also there, maybe not in this class. So, we will load it again. Okay, so we have been to that directory. Now we will load that data. We have seen that data before. Is this file? We need the header. Header equals to true. We have the header lines, and as I showed you, we can check this data in such a nice way, right? This is much nicer than looking into the console because this looks pretty bad. Okay, so this is how it looks like. You can scroll and you can see the data. Okay, so these are the gene names, the first column, and then we have these different parameter values. And let's say we want to, what we want to do is we want to take this analysis a bit toward more complex, right? And how do you do that? So what we'll do is, let's say we'll classify or separate the data. We'll use the DMYPD data, okay, for plotting. But before that, we want to do this, compare these two classes, right, based on the parameter value data box. Okay, so as you will see, and I have mentioned before, it says data box can take only 0 or 1, right, whether you have data box in the promoter or not. So what you, for example, let's say we want to test whether having data box matters for noise or not. Okay, so what we will do is we will first separate out the data into two classes, right, and then we will test those classes. So this separation would happen based on the value of the data box parameter. Okay, so let's do that. This is slightly more complex to compared to what we have been doing until now. So let's do that, and here I have the code, right. So this is how we will work, right. So no data box, let's say we assign, again we can use any assignment parameter. This is from the data, okay, and I mentioned, right, when we are discussing data structures, etc., and as well as vectors, what we can do is we can use this command called which data box equals to 0, take those rows and store in this no data box frame. Okay, so what we have is which okay, so that's the command. So which means, this means test the data box value, right. If it is 0, for those, take those rows and put in the no data box and create a new data frame. Okay, and similarly we can test for data box equals to 1 and put in a separate data frame, which is yes data box. Okay, so view, let's say we can check this data, just one of them, let's say, and you see this is a new data frame now that has been created. You can see this very clearly is here is the data box value is 0 for all of them. Okay, and similarly you can see the other one where the data box value would be 1 for all of them, right. So we have separated out this data frame based on one parameter. So this is something that's possible in R. Okay, and you can do this for any parameter. It doesn't always have to be equal to 0 or equal to 1. You can also separate out, for example, greater than some value cutoff and less than some cutoff. You can do that as well. So this is possible in R. So we are splitting the data frame based on some conditions. Now once we have split this, the command that we use is called box plot. Okay, the box plot command okay, and this is how this whole thing works. Oh sorry, yeah, so we cannot plot the full data frame, right. So we have to use, let's say, DMYPD, right. We want to compare noise values, and this is what we get. Okay, so the first one is the no data box and the second one is yes data box, and we are checking the noise values. We can make it nicer as before, and this gives us labels 1 and 2. So we want to make it clear that this is no data box and this is yes data box. Okay, and for that, first we have to, when you generate this plot, we have to say x axt is equal to n. Okay, so these labels are gone. Okay, these numbers are gone because we will add them later on. What we can also do, for example, is say we can, of course, the standard practice which is there is 1. Okay, y label we can give, right. So these are standard which we have done already. So you know, Okay. And then we can add the axis. Okay. So, the other thing we can also do is we can make them colored, right. We can use two different colors for these box plots. Okay. So, we can say color is equal to, so we need to define two colors, right, unless they will be colored in the same way. So, for doing that we have to define as C, right. So, the first one would be, let us say, red, let us say, right, and the second one we want to use blue. Okay. And we are using the same format, this RGB, and we have defined two colors, right, for this plot. Okay. And then we end the plot. Okay. So, you see, like, we have added the colors. Okay. So, this is okay. What we want to do now, we want to add these labels on the side. Okay. And this is the command. Okay. I will just explain in a moment. So, axis side equals to 1. So, on this side add labels. So, these two labels are required. If you have 3 labels, if you have these 3 box plots, right, or 3 such values, right, distribution of values, you have to give 3 labels. Okay. And you have to also mention where they should be at C equals to at C 1 2, right. So, here the values were 1 2. So, at C 1 2 you have to add these labels. Okay. And what would be the size of the labels, etc., 6 dot axis, right. So, that will be the size of the labels. Okay. So, this is how it looks like. Okay. And we have added the labels. So, this is how we can generate this very customized plots. What is not so good is that we cannot see the data points, right. As I said, sometimes you would like to see the data points, right, where the data points are. You see some of the data points, but these are mostly outliers, right. So, these are actually out of this IQR. So, this is the median and the interquartile range. So, they are outside. So, we are only seeing those data points, but we want to see the distribution of data points here as well, right. And this is something we can do in here, and that is what we are going to do now, is using this strip chart function. Okay. So, this is the strip chart function that we are going to use, but before we can use the strip chart function, what we have to do is we have to generate this box plot, but with this command called outline equals to false. Okay. So, what happens when you use this outline equals to false command is that it removes all these points that are beyond this IQ or interquartile range. Okay. So, if they are beyond this, right, they will be removed, but then it also changes the scale of the axis, right. So, we want to maintain this y axis scale, right. So, we want to say y limit is C, these values are around -5 10. So, let us use -8 and maybe, and then the upper limit is 60, let us say, right. So, we keep that scale. Okay. So, -10 to 60. Okay. We can use the axis command anyway, it does not is the same, right, the axis command labels. So, these are given. What we want to do now, we want to add these data points on top of it, and the command is called strip chart. Okay. So, how does this work? So, these are specific parameters that you have to use. Again, how do you know these parameters are required? You can search for help, etc., and look for this what are the parameter values, and these are all explained very, very nicely in strip chart. Okay. So, for any of these functions that we are using, you can go to their help pages, right, the manual, and you can see what these functions do and what are the parameters they will take, etc. Okay. So, here for the first one we want to add this. So, we have to add these points one at a time. Okay. First for the no data box data and then for the yes data box data. Okay. So, no data box data dm ypd, right. Then we have to say this is vertical equals to true, right. Then we have to say method is zeta. So, I will show you in a moment what zeta means. Zeta means the points will be spread around, otherwise they will be all overlapped with each other. Okay. So, zeta means they will be spread around little bit, right, so that we can see the concentration of the density of the points in here. Okay. This is it, and then probably we also, yeah, we need to add this add equals to true, right. We are adding to this plot, we do not want a separate plot, and then we also have to say where we want to add this. So, this is no data box data. So, we want to add this at 1, right, and I believe we need to add the colors, right, unless you want all black colors. So, for the no data box we are using red color. So, let us keep the same color, and okay. So, we have not used the other one, which is the PCH. Okay. So, we have to, let us try the PCH, right, let us try PCH 20. This is how it looks like. Okay. Maybe we want slightly lighter colors, right, and so for that we need to change this one, right. Okay. This is how it looks like now. You can see the concentration of the points with the zeta, etc. We have to do the same thing for the other one, right. So, this will be ES data box data, right, particle true method zeta, this will remain the same, add equals to true at the second point, right, in the second column here. So, this column color will also have to change to blue one, and this is what we get. Okay. So, you see this, now we have generated this, and we can also now add the axis labels, and this is the complete bar plot. So, this looks much nicer, right. We can now see the data points as well, how the data points are distributed, etc., and we can see the median values, IQR, etc. We can compare them as well. Okay. We can do this for, so box plot is very useful because we can do this for multiple plots, right, multiple parameters. Here we are comparing only 2, right, no data box, yes data box. You might have multiple classes and you want to compare them, right. In that case, the y axis is showing the expression noise distribution. Okay, so this is fine, but this is also a bit cumbersome, right. So, for example, the data density is very high here. For that we use the Bio plot. Okay, so in the next class we will talk about the Bio plot taking the same data, and then we will also talk about some more advanced visualization techniques. Okay, so with that I will conclude. So, what we have seen in this class, so we have talked about box plot and we have completed the mean SD plot, but we have not seen the Bio plot yet, and in the next class we will be talking about the GG plot too. This is now we are getting to more complex visualizations, and again here we have done some hands on, and we will continue that in the next class. Thank you.